Good evening, ladies and gentlemen of the biochemistry community. I am Michael Robinson, coming to you live from the heart of Thomas Merton. We have breaking news about two sister proteins, hemoglobin and myoglobin, who were separated at translation but have been recently reunited in an injured patient. But first, let's explore the structure of hemoglobin and myoglobin. Hemoglobin is a tetramer composed of 4 globin molecules that interact via non-covalent bonds. The two alpha chains are composed of 141 amino acids, and the beta chains are composed of 146 amino acids. Each globin protein is made up of 8 alpha helices. The globin groups interact with the 4 prosthetic heme groups that each contain iron and bind to an oxygen molecule. There are 2 allosteric structures of hemoglobin, the relaxed structure R, or oxyhemoglobin, which has a greater affinity to oxygen, and the taut structure T, or deoxyhemoglobin, which has a lower affinity for oxygen. The affinity for the R structure becomes greater after each oxygen binds to the iron atom until the hemoglobin is fully saturated with 4 oxygen molecules. It is through the cooperativity of this molecule that is able to carry oxygen from the lungs to the muscles and carbon dioxide from the muscles to the lungs. Now that we've learned a little bit about hemoglobin, let's go to my co-anchor, Carly Johnston, to talk about myoglobin.
Thanks, Mike. Myoglobin is a single-chain globular protein made up of 8 alpha helices connected by loops. The 154 amino acids that make up the single polypeptide include histidine residues at the 94th and 65th positions, which allow the protein to bind to oxygen at the heme group located in its hydrophobic core. This heme group is an iron-containing compound that acts as a prosthetic group in the protein, without which it would not be able to function. A fun fact about myoglobin is it is the first protein to have its 3D structure revealed, and now we'll be going to Allie Paz with the patient.
Thanks, Carly. As the patient was working out, the hemoglobin in the blood near his lungs was picking up oxygen. The hemoglobin has an increasing affinity for the oxygen molecules, meaning that once the first molecule is bound to the hemoglobin, the second one is more likely to bind, followed by the third and fourth molecules. Once fully saturated with 4 oxygen molecules, the hemoglobins flowed throughout his body in order to deliver oxygen to the muscles. When the hemoglobin deposited the high concentration of oxygen molecules in the lungs, the hemoglobin was in the relaxed, or R structure, that has a high affinity for oxygen. However, as the hemoglobin goes into the low oxygen areas, it rotates to the tense, or T structure, which has less oxygen affinity. Once one of the oxygen molecules detaches from the T-structured hemoglobin, it becomes more likely that the next oxygen will detach. Therefore, the oxygen detaches from the hemoglobin in low oxygen environments, like the muscles. Then, the hemoglobin takes up the carbon dioxide and carries it to the lungs, where the carbon dioxide is expelled from the body. The oxygen molecules travel from high concentration to low concentration through simple diffusion, bringing oxygen into the muscle cells. This is where the myoglobin comes and binds to the oxygen molecule. The binding causes a structural change, and the myoglobin does not release the oxygen unless extreme conditions of high carbon dioxide levels in the blood and a high concentration of lactic acid in the muscles occur, much like what happens during exercise. The myoglobin can bind to one oxygen and then transport into the mitochondria so the muscle cell can use it in cellular respiration to produce energy. As the patient was exercising, the muscle tore, and the myoglobin was released into the blood, uniting the 2 globin proteins in the bloodstream.